diseases caused by viruses. Common cold and polio are two diseases that are caused by viruses. Rhinoviruses cause the common cold. Poliovirus causes polio. Rhinovirus are frequently encountered, and we have various different strains of rhinovirus, and they're essentially all causing the same kind of disease, with sneezing, congestion, difficulty breathing, sometimes ears get infected, eyes are itchy, definitely very uncomfortable. And because there's so many types of rhinovirus, it's been virtually impossible for scientists to create a vaccine that would reduce the occurrence of the common cold. And of course, the other problem with having a cold, which is a viral infection, would be the occurrence of secondary bacterial infections, adding misery on top of already being miserable. The other kind of virus in the same family, which is Picornaviridae, or what we would generally call picornaviruses, are the polioviruses. Polioviruses cause polio. It's a type of virus that's ingested, whereas rhinovirus is one that would be inhaled or could be airborne. And with poliovirus, this virus attacks the nervous system. And so typically we would see an array of neurological symptoms, leading to the worst possible neurological symptom, which is paralysis. We might have paralysis of peripheral limbs, especially the legs, and in some cases, almost complete body paralysis. In the 1930s and 40s, it wasn't unusual to find people avoiding public ponds, municipal swimming pools, lakes, rivers, streams. Lots of children especially would not go out in the summertime to use those waterways for recreation or swimming, because it was fairly well known that that type of exposure could expose a person to the cause of polio. And sure enough, a good example is Franklin Delano Roosevelt, President of the United States. He contracted polio by swimming in a lake that had poliovirus in it, and did become paralyzed, and in fact his legs were paralyzed for the rest of his life. If you see in the middle picture an illustration of an iron lung, once a patient contracted polio and essentially developed paralysis of the diaphragm and wasn't able to breathe on their own, they would be put in this large metal container, and the iron lung would breathe for the patient. The patient would essentially spend most of their life in this container. There's typically a little mirror that they could use to see everyone else and watch what was going on, but they were pretty much limited to living in this iron lung. Now we have better means of assisting patients with respiration today, so we don't use iron lungs today, but we still have occasionally patients who do develop polio. Now if we go out into the water systems and various estuaries here in the United States, take a water sample, we can find poliovirus. Now typically it's not what we would call the wild type poliovirus, or the original poliovirus that causes polio, but instead the vaccine version of the virus. There are two types of vaccines that help us to prevent polio. One is called the Salk vaccine, which is heat-killed and is injected, and the other is called the Sabin oral poliovirus vaccine, which is attenuated living virus. Attenuated means weakened. So when Dr. Sabin created his oral poliovirus, the intention was to essentially expose our digestive system, which is the most likely portal of entry of this virus. And by exposing our digestive system to a weakened version of this virus, or the vaccine-grade virus of polio, it allows our bodies to develop active natural immunity at essentially what would be the portal of entry, through the lining of the digestive tract. Well, of course, any extra virus that's in the digestive tract after receiving this vaccine winds up in the toilet, gets flushed out to the wastewater treatment plant, eventually gets processed, but the processing in the wastewater treatment plant does not remove most viruses. So what gets dumped back into the lakes and streams are the vaccine-grade poliovirus. Now, occasionally there are some complications with every kind of vaccine, but after thorough investigation, we ideally produce vaccines that have greater benefit than the risk. It's more difficult and more risky to actually get the disease, and that's definitely true with polio. Another important group of viruses are the orthomyxoviridae, also generally called the orthomyxoviruses. These are negative single-stranded RNA viruses, and the example that we're going to pull out of this particular family would be influenza virus. There are actually 3 different types of influenza viruses. We have influenza virus A and B, and influenza virus C. Now, influenza virus C really doesn't cause us a problem that much. The 2 influenza viruses that do give us concern are influenza virus A and B. So we have type A and we have type B. They're not combined, they're 2 different types. Each influenza virus group produces spikes on the surface of the envelope of the virus. We have hemagglutinin spikes, which are illustrated in red on the image on the left-hand side of the little cartoon drawing of the different kinds of influenza viruses. We also have N spikes, which are illustrated on the drawing as sialidase spikes. Neuraminidase, if you look at the word ending with ASE, automatically tells us this is an enzyme. So this is a chemical that's going to facilitate some kind of chemical reaction. It's a biological catalyst. And in fact, the hemagglutinin and the neuraminidase spikes are used for attachment. If you can see in the cartoon drawing, where there are items, these little round spheres that are sticking away from the surface of the cytoplasmic membrane of the cell that this virus is targeting, and the H and N spikes are attaching to these little structures sticking out from our cells. This is the first step, the attachment of the virus to the cell that it's going to infect. And of course, the first step of any good viral infection has to be attachment or adsorption. And sure enough, these spikes facilitate or help influenza virus to do just that, to get a good grip or attachment to the cell it's trying to get into. With influenza virus in general, for the orthomyxoviridae, we find that disease can occur year-round, especially in groups of individuals or people that are in large groups in confined spaces, such as apartment buildings, dormitories, or military barracks. And especially when we have to all go inside and are more likely to stay inside in large groups because of cold weather. So we often see an increase in the prevalence or the number of cases of influenza, especially in the wintertime. Typical cases of influenza nowadays we usually see in the very young or the very elderly, because those individuals often have either compromised immune systems in the case of the elderly, because their immune systems are sort of wearing down or not as vibrant or as powerful as, let's say, a 20 to 30-year-old. And with children, their immune systems are just developing. So again, they're less likely to be able to withstand the challenge of the viral infection. We find that the incubation period for influenza viruses today is about 1 to 4 days. So yes, it is possible for someone to sneeze or cough or you get exposed to the virus one day, and the next thing you know, 24 hours later, you're not feeling so well. We find that the worst symptoms occur after about 5 days, With children, it can be 10. And as most of you well know, if it's influenza, sometimes it can hit fast and furious and make you very sick very quickly. But usually it's limited to perhaps 10 to 14 days in length of illness. That's in contrast to the common cold, which can often linger for weeks with difficulty breathing and the typical symptoms that we associate with the common cold. So influenza and the common cold are often confused. Influenza typically hits quickly and is a much shorter duration than a rhinovirus infection. We see the highest rate of infection due to influenza in children, though I do think in recent years that has shifted a little bit to the elderly. If we're taking a look at symptoms, in addition to stuffy nose, maybe a scratchy sore throat, and similar symptoms to what we might see at the beginning of a common cold, which is rhinitis, inflammation of the nose, we might also have a non-productive cough. Sometimes that's a dry cough. We just don't feel good, that's the malaise. Sometimes with muscle ache, that's the myalgia. Headache, maybe even a fever. If we're looking for treatment, we might simply have bed rest, fluids, and lots of good homemade soup if possible. And of course, we do have a few antivirals that have been very effective, though with limited availability and oftentimes limited access, depending on how much has been produced and where it's been essentially provided to different pharmacies. If we take a look at diagnosis, we could do viral cultures. If you go back to the section where we talk about sort of essentially diagnosis or identification of the presence of a virus, we can take samples directly from a patient and look for the virus in essentially the swabbings or material collected from the patient. We can also do immunoassays, looking for evidence of the virus being present. And the worst case scenario would be complications due to a secondary bacterial infection. So we essentially add misery on top of misery with two infections, both bacterial and viral, in the same patient at the same time. You might recall that we mentioned that influenza viruses have spikes. We have hemagglutinin spikes and neuraminidase spikes. There have been 16 different types of hemagglutinin spikes discovered so far and 9 different types of neuraminidase spikes. With all the possible combinations of H and N spikes, it's possible to have 144 different subtypes of influenza virus with all of the possible combinations of H and N spikes. The common human subtypes are H1N1, H1N2, and H3N2. Those are further subdivided into even more well-defined strains within each subtype. We do find influenza A in birds, pigs, humans. In fact, a variety of domesticated animals can have influenza A, either causing them infection or simply found in the animal and passed from one animal to another. Influenza B is found only in humans, often in long-term infections, less likely to cause epidemics in comparison to influenza A, does not cause pandemics, and has strains but not subtypes. So influenza A truly is the pandemic version of influenza, more likely to cause worldwide infections or epidemics than any other type of influenza. Influenza C only causes infections in humans. These are individual cases and does not result in epidemics or pandemics. influenza A, we can have domesticated birds, wild birds such as ducks, pigs, and humans as essentially mixing vessels for various strains and subtypes of influenza A. We see that the virus can adapt to the physiology of the different organisms. Once inside the different organisms, we have a reassortment of the H and N spikes. We even have adaptation to various organisms, animals, people. Very interesting kind of complex exchange of virus from one organism to another. Influenza viruses, influenza A especially, but influenza B can also have this occurring, often have what's called antigenic drift. These are small gradual changes in the structure of the H or N spikes because the H and N spikes are made out of proteins and as such are susceptible to mutations. Oftentimes it's point mutations that are found, these small, slight little changes in the amino acid sequence or arrangement of the H or N spike. Another feature that's often seen in influenza A is antigenic shift. With antigenic shift, we often see a dramatic change in the spikes that allows the virus to essentially what we call jump species. Perhaps this was a significant change in the amino acid sequence in the H or N spike and allowed the virus to more easily be transferred from an animal to a person. The little cartoon drawing at the bottom, I think, demonstrates the difference between drift and shift very well. Notice that if we're talking about antigenic drift, the spikes that are on the surface of the virus are very similar to what was on the surface of the original virus, those little pointy arrow-like structures. But with antigenic shift, we see a dramatic difference in the construction or the shape of the spike. So the antigenic shift, a significant change, allowing the organism or the virus to jump species. Antigenic drift, slow, small, gradual changes that can also eventually lead to the virus being not nearly as virulent or as infective as it might have been when it first started to develop. Here are a couple more illustrations or additional illustrations of antigenic drift. Notice that we see small, subtle changes in the configuration of the H and or N spikes. With these subtle changes, it will either allow the virus to attach to cells that they weren't able to attach to before, or maybe allow the virus to resist the effect of certain immune factors. But again, these are small, subtle changes if we're talking about antigenic drift. Here are some examples of various flu vaccines that have been used in the last few years. Notice that we have various combinations of H and N spikes that have been used. Oftentimes scientists get together and a year ahead of time try to predict as best they can what's the most likely strain or subtype of influenza virus that will show up the next year and begin manufacturing the appropriate vaccine to give the most individuals the most protection during that next epidemic-prone season. In this illustration, we see the possible ways that antigenic shift can occur. Oftentimes, due to a particular cell being co-infected with perhaps two different strains of influenza virus, coming from perhaps two different sources, like from a bird and a human, or a bird and a pig. Notice at the bottom of the illustration we have what's called reassortment. We have sort of a mixing and matching of genetic material from the two different viruses, leading to the presentation of different combinations of H and N spikes. It can happen if we have the virus passed right from a bird to a person, if we have persons passing the strain to birds or pigs, if we have, again, co-infection with two different kinds of influenza strains, mixing their genetic material in human cells, or we can have essentially genetic change of a bird strain of influenza A when it's introduced into other birds or other intermediate hosts like pigs. Just all kinds of interesting rearrangement and swapping of viruses and swapping of genetic material, leading to the presentation or the emergence of new various combinations of H and N spikes. Now, why should we worry about influenza? Well, the problem with certain strains of influenza, and we do group or identify different strains of influenza by the type of hemagglutinin and neuraminidase spikes that are produced. They do vary. And there are certain H and N combinations of influenza viruses that cause epidemics, or even pandemics, where we see worldwide epidemics, same disease, on multiple continents or involving multiple countries. In fact, influenza epidemics and pandemics seem to cycle. Various timelines, if you take a look, we've got 1918 was the Great Influenza, the Spanish flu, when so many millions of people died. 1957, 1968, frequently because of the cyclic nature at which influenza causes epidemics and pandemics, medical communities can often predict or estimate when the next great epidemic or pandemic will occur. And in doing so, they try to prepare ahead of time to prevent the huge loss of life that's occurred in the past. In 1918, we started to see the emergence of a new strain of influenza that caused a high degree of mortality, especially in young individuals, particularly young men. Lots of folks were affected, realized that children and elderly also got the disease, but it was more likely to be 20 to 30-year-olds, both men and women, and in large part because we had a lot of soldiers grouped together in military camps, overseas, in various areas to fight the war. World War I was occurring at the time. That could, in fact, have been part of the reason that we saw a significant mortality among the troops because of the influenza. In fact, just a few years ago, about 7 years ago, that information was finally declassified, that there were more individuals, especially soldiers, who died of the flu, an infectious disease, than died of battle wounds. And it was kept quiet, or kept pretty much as a military secret, because we really didn't want our adversaries knowing that most of our soldiers were either sick or dying due to a disease and unavailable to fight in the war. Now, it took about 9 months for this pandemic to circle the globe. Well, this particular pandemic took 9 months to circle the globe. We still do have fairly modern modes of transportation. not quite as fast as today, but we had airplanes, boats, trains, automobiles, and so that did contribute to the rapid transmission from person to person of this particular phenomena and the global expanse that it hit and affected people on multiple continents and multiple countries. The estimates of number of people who died go as low as 20 million and as high as 100 million. We find that in human history, when we have a lot of people dying all of a sudden, we fairly quickly lose count. The agencies that are responsible for monitoring the health and wellness of different countries, the public health agencies, can't keep up with the death rolls. Morticians, clergy, folks that are supposed to help to take care of the dead, bury the dead, can't keep up with the high numbers. And that's particularly why we see a huge range of mortality for the great influenza of 1918. It's been cited to possibly be the most devastating epidemic, even more than some in previous human history. Between 1918 and 1919, it's estimated that more people died in that single 9 to 12 month period of time than died in the 4 years in Europe of the Black Death. Now, granted, remember the numbers are pretty much vague estimates, but that's a significant statement. The reason that we didn't see such a dramatic social and political change that was seen with the event of the Black Death was because in the Middle Ages, and particularly in Europe, the scene was set and the stage was set for some dramatic political, environmental, and social changes with so many people dead and dying. And the relative percentage of people dead in the Middle Ages was actually much higher because the total population of folks that were alive was much smaller than what we saw at the beginning of the 20th century. This great pandemic, or the great influenza of 1918, is often called the Spanish flu or La Grippe, because it was originally thought to have started in Spain. But in fact, it really started in the United States in Kansas at a military barracks where a soldier contracted the virus and before he showed signs or symptoms was essentially passed off or shipped off to Spain. It's at that point we start to see essentially the emergence of this particular strain of influenza, which as it turns out was an H1N1 influenza strain. We're going to see H1N1 again when we talk about a more recent pandemic of influenza. Now, various epidemics or pandemics of influenza have occurred. If you take a look at 1957, what was called the Asian flu, it was a type A, H2N2 avian flu. In fact, most influenza does pass from birds to pigs to humans. There's a typical sort of cycle or transition from one animal to another, and every time this influenza virus or these influenza viruses enter into a new host, there's a little bit of a rearrangement or change in perhaps the virulence, which means the severity of the symptoms that they cause, and possibly some features about the spikes. Well, as it turns out, the 1957 Asian flu caused a mortality rate of about 1 to 4 million people, but was limited in its spread because by this time we have some good influenza vaccines that can be used to prevent folks from getting the flu. And so this is a good example of the effectiveness at the dramatic reduction of morbidity, which would be sickness, and mortality, death, due to a disease organism or pathogen, like the influenza virus. In 1968, we have the Hong Kong flu. Oftentimes, these different epidemic or pandemic strains of influenza are labeled perhaps based on where people think the original index case or starting point was for the epidemic or pandemic. Sometimes accurate, sometimes not. In any case, the 1968 Hong Kong flu was a different type of influenza A. It was an H3N2. We have, again, an avian influenza. It killed roughly 1 million people. Mortality was much lower because of the much more successful use of influenza vaccines. And eventually we do start to see the use of antivirals in the 1990s. But in 1994, with antivirals used to treat various cases of influenza, we see only a 1% drug resistance among the different viruses that are causing influenza. By 2003, with increased use of the antivirals, we see an increase in drug resistance among samples taken. By 2005, virtually 91% of all of the samples taken and evaluated for drug resistance were in fact resistant to the effect of commonly used antiviral drugs such as amantadine and rimetadine. The more we use medications to treat various kinds of microbial infections, including viral infections, the more likely we are to encourage the growth of the drug-resistant strains. It's an unfortunate consequence of using these medications, but sometimes it's better to use the medication and survive the illness than not use it and risk mortality because you didn't use it. 2004 to 2006, a slightly new subtype of an H5N1 type A influenza virus appeared, often called HPAI H5N1, because it was a strain that was occurring in birds, domesticated birds like ducks and chickens, that appeared to be undergoing antigenic shift and moving easily from birds to humans. Now, there really weren't that many cases of human influenza A, HPAI H5N1, but public health authorities certainly didn't want to risk this virus escaping and causing widespread epidemics or pandemics. So fairly quickly, there were efforts to curtail the spread of the virus among domesticated birds, which would include chickens and ducks, and many populations or farms had to eliminate or kill their animals to prevent the spread of infection among the birds, because the birds also get sick, and they had to monitor the human populations fairly closely, because these are areas of the world where people often live in very close proximity to their animals. So sure enough, by the time we get to 2007, the epidemic or pandemic that many public health authorities had predicted would occur never did. And probably because of all the efforts that were undertaken to dramatically prevent the spread of this virus. March 2009, in Mexico, and essentially cases of influenza started to show up in that late spring of 2009. Mortality rate was a little higher than would be expected and a little worrisome to the public health authorities there, around 25%. That's pretty high in this day and age. So an alert went out, and Eventually, within a few weeks, the virus had entered into the United States. In April, we had our first case on April 15th in one state. Within another week or so, we had about 5 to 6 cases, a few of them showing up in essentially Texas and California. Within another couple of months, April, May, and June, by June 19th, there were 21,449 cases in the United States, and it was in all 50 states. In fact, the progression and the transmission of the virus within those several weeks between April 15th and June 19th, it was interesting to monitor and watch almost on a daily basis the spread of the virus from those gateway states of California and Texas to the rest of the 50 states, including farther north in northern Canada and Alaska. Well, sure enough, the World Health Organization, by the time we get to the end of June, has declared a pandemic. We find that this particular virus has moved through not only numerous countries, but numerous continents. As the pandemic proceeded, and scientists began to determine the subtype of the influenza A virus that was causing this new pandemic, it was discovered it was an H1N1 strain, or subtype. Not quite as virulent as the H1N1 from 1918, but certainly perhaps as contagious. Fortunately, by the time the virus entered into the United States, perhaps through some kind of recombination, it wasn't nearly as virulent as it had been in Mexico or Central America. The mortality rate was much lower for the remainder of the pandemic, though I did work with students who had patients who had this particular strain of influenza that year, and their patients were very sick. So you definitely wouldn't want to be one of those individuals who accidentally got infected with the H1N1 of 2009. This is just a summary of some of the unique features of the various pandemics or epidemics of influenza that we've just highlighted, where we have the Spanish flu of 1918, the Asian flu of 1957, the Hong Kong flu of 1968, the Asian flu H5N1, and the most recent H1N1 of 2009, which for a while was called a swine flu, but really was misnamed. And we really just should refer to it as the 2009 H1N1. Now, one of the reasons we're spending a little more time than usual discussing viruses, and particularly influenza virus, is because its capability of causing epidemics and pandemics, the high mortality that can be associated with those infections, and the importance, depending on where you wind up working, of understanding how this virus is acquired and the symptoms associated with it. And even folks working in laboratories have to pay attention to their microbiology class. And what happens when you don't pay attention to your microbiology class and remember what you learned? In October of 2004, an agency called the College of American Pathologists, which assembles kits of actual specimens from patients, sends them all over, essentially North America, for testing and possibly international testing to verify the proficiency and accuracy of laboratory facilities testing all types of specimens. And that also includes virology samples. So these test kits that are sent to the clinical hospital laboratories or any of the clinical reference laboratories to essentially test them on their ability to do their job and do it accurately and well. Unfortunately, the test kit that was sent out in 2004 contained an pandemic strain of H2N2. So whoever essentially assembled or manufactured that kit probably didn't remember what they had learned in their college microbiology class. Between October of 2004 and February of 2005, these kits were sent out to various labs all over North America. Fortunately, some of the testing facilities recognized what it was. About 3,700 laboratories received these specimens, and several of them realized early on that this was a very dangerous strain of influenza to put into a kit. Sort of let College of American Pathologists know, or CAP, know about this problem, and the test kits were recalled. The greatest fear was that somehow the virus would escape a laboratory facility. The fact that it didn't escape worldwide was a miracle, perhaps. It did escape a high containment lab in Canada, but was quickly contained, and the worldwide epidemic or pandemic that was so highly feared was prevented. With this next group, we have unusual viruses that have a double capsid and are double-stranded RNA. Now, typically, when we find a virus that has two coverings, it's a capsid in an envelope. Well, these viruses instead have two capsids, an inner and an outer capsid. Very unusual. They also have double-stranded RNA as their genome. Now, usually when we find a nucleic acid in a double strand, it's going to be DNA. Usually RNA is usually single-stranded. So, a couple of very unusual characteristics for these example viruses. The first example virus would be the reoviruses. Reovirus are often associated with tumor regression or tumor lysis. The medical term for tumor lysis is oncolysis. We often describe the reoviruses as oncolytic. In fact, the last few years, they've been used in the manufacture of an oncolytic drug called reolysin. You can see the reference for reolysin on the slide from 2008. There's also an illustration in the top photo of the little mouse who in the left hand image has a fairly sizable tumor on his left hind leg. After treatment with reovirus, the tumor is all but gone. Another example of a double capsid, double-stranded RNA virus would be the rotaviruses. Rotaviruses can cause upper respiratory tract infections, or URTI, and gastrointestinal tract infections, GITI, especially in young children. We particularly worry about GITI, or diarrhea, in young children because they can lose a significant amount of body fluid and body volume fairly quickly with really severe, serious cases of diarrhea or dysentery. So we often in the laboratory have a means to screen or determine if the patient, especially a young toddler or young child, has a rotavirus infection. Retroviruses are positive single-stranded RNA viruses, and if you launch this slide in PowerPoint, watch as it runs through an animation that goes through all of the stages of viral replication and viral life cycle for retroviruses, starting with adsorption, then going to penetration, showing the formation of the provirus, which is essentially the insertion of a DNA copy of the viral genome into the host chromosomes. And then the virus essentially commandeers or directs the synthesis of viral proteins and nucleic acids with assembly, maturation, and release of the new viral particles. The animation even shows the formation of viral spikes, which we see on the surface of this little cartoon drawing. These retroviruses include viruses called HTLV, human T lymphocytic or lymphotrophic viruses. These viruses, HTLV viruses, tend to infect white blood cells that we call lymphocytes. These lymphocytes can be found circulating in peripheral blood, or they can be found in lymph nodes or various lymphoid tissues. HTLV 1 and 2 are frequently associated with leukemias and malignancies like lymphomas. HTLV 3 also has another name, which is HIV-1. HTLV-3/HIV-1 is the virus most associated with acquired immune deficiency syndrome, or AIDS. For any of these viruses, they gain entry into the white blood cells, or the lymphocytes, form that provirus construct, and result in what's often described as a slow viral infection. In the original infection, you don't see many viral particles being produced. The more long-term the infection, the more viruses are released. And you can see in these color illustrations of the transmission electron micrograph in the lower left of this slide, all these sort of artificially color-enhanced little dots emerging from the surface of the cell are newly released viral particles. So this enveloped virus is one that can cause various types of chronic, long-term, often high mortality, life-threatening diseases like the leukemias, lymphomas, or even AIDS. The enveloped double-stranded DNA viruses, example that we have to use, are herpes viruses. These viruses are a little bit different because they cause latent viral infections. We'll see in another slide or so, these viruses gain entry into our bodies and essentially never leave. We have herpes groups alpha, beta, and gamma, all capable of causing a wide variety of different types of infections. In fact, we'll find that herpes viruses can be transferred during pregnancy from mother to child and result in birth defects. If you recall talking about TORCH, T-O-R-C-H, which is an acronym that stands for the various microorganisms that are frequently responsible for congenital birth defects, or that are teratogens that can result in damage to the fetus during pregnancy. The TO stands for toxoplasma, the R for rubella virus, the C for cytomegalovirus, which is in the family of herpes viruses, and the H for herpes virus itself. Can be passed from mother to child, causing serious damage. In the alpha herpes group, we have 3 viruses that are very common and very important. We have herpes simplex 1, which causes the common cold sore. We have herpes simplex 2, causes genital herpes, and varicella zoster, which causes chickenpox and shingles, and a condition called St. Anthony's fire, which is essentially shingles. Herpes simplex 1 and 2 and varicella zoster all have the ability to undergo latency, which is a form of inactivity because the virus enters onto the surface of mucous membranes or skin, but typically mucous membranes, attaches to nerve fibers, travels down the nerve fiber to a ganglia, and establishes latency or becomes dormant and resides in that ganglia for many years, possibly. Could be as long as a lifetime, could be as short as just a few days. The reactivation or the reawakening of this virus from latency to becoming an active infection is varied. What can cause that reawakening could be changes in diet, temperature, stress. There is, as far as I know, no one event or one factor that essentially breaks latency and causes the virus to once again become an active infection. But in any case, the reawakened virus will travel back down the nerve fiber, back to the surface of the mucous membrane, or oftentimes it's an interface between mucous membrane and skin, such as around the mouth, and cause those vesicles that can then rupture and cause those scaly, oozing lesions that we associate with, let's say, the cold sore. Now, simplex 1 and 2, simplex 1 typically is found causing cold sores, and simplex 2 with genital herpes, but they're not mutually exclusive. You can find simplex 1 causing genital lesions. You can find simplex 2 infecting the oropharynx. Now, with varicella zoster, which causes chickenpox, It also can undergo latency, and then in later years, especially with the elderly, reemerge, causing shingles. The photo in the middle of this slide shows an individual with shingles. Because of the sort of localized regions that ganglia and the nerve fibers attached to the ganglia serve in the human body, you can see that his shingles has a very regional appearance of symptoms. Those vesicles and those red oozy lesions are essentially only seen in the left upper quadrant of his face. Now, years ago, before we had a really good vaccine for varicella zoster, which has limited the occurrence of chickenpox, it wasn't unusual for grandma and grandpa to have shingles and transmit that virus to the kids, who then developed chickenpox. And chickenpox was so highly contagious before the vaccine, it wasn't unusual for mothers, if someone had a child who developed chickenpox, mothers would just get all of the kids together in one home, expose them all at once in a chickenpox party, so that all the moms or mothers in the neighborhood could help one another, and everyone would essentially get through that process of having the chickenpox early on and all at once. Now, it's really not desirable because there are some adverse effects of chickenpox. It can cause blindness, damage to ears. You'll find that these pox-like lesions can show up everywhere. So, truly, the fact that the chickenpox vaccine or varicella vaccine is very effective and readily available, it's a good idea to take advantage of that availability. Now, what if somebody manufactured a vaccine that could prevent people from dying of cancer? Would you use it? Should we use it? It's estimated that 15% of all human cancers are due to certain types of viral infections. Not just HPV or human papillomavirus, but a wide variety of viruses. Human papillomavirus, or HPV, is just one good example. It's a naked double-stranded DNA virus, part of the family that we call Papoviridae, and is the cause of warts. All kinds of warts. We have warts that show up on fingers, warts that show up on the bottom of your feet that we call plantar warts. We even have genital warts, all due to infection with viruses from this group, the papillomaviruses. Another name for a wart is a papilloma. Here's an example on this slide of the progression and the pathology associated with HPV infection and the association and cause of cervical cancer. When HPV infects the cells on the surface of the cervix, it results in what's called transformation of those cells. If you go back and look at some of the cytopathic effect that can occur with certain types of viruses, you'll recall transformation is one of those CPE. The cells no longer function normally nor appear normal because of the virus that has gained entry and started to cause changes inside of the cell. The cells take on different appearance, and of course, the more this particular change progresses, we can see that we have the change from normal cervical cells to cervical neoplasms, which are called new cells or new-like cells, which means they're no longer normal, then all the way on up to cervical carcinoma. So this particular illustration does try to show essentially the changes in the nucleic acid of the virus, as well as when we have integration of the viral DNA into the nuclei or the chromosomes of the cells in the cervix, which then lead to various stages of pathology or abnormal changes. two more examples of the types of abnormal changes that result when cells are infected with HPV. On the left, we see an overproduction of structures that we call centrioles, and also a change in the distribution of DNA inside of the cell. The DNA is stained red, and the centrioles are stained sort of apple green. And when we have the HPV virus infecting the cells, some of the pathological abnormal changes that are as a result of HPV infection means that we see over duplication or production of these centrioles. So we see more of them in the HPV 16 E7 cell infected on the right versus the control on the left. And if you take a close look at the larger image of the HPV 16 E7 infected cell on the right, you'll notice there's lots of DNA speckled throughout the cytoplasm of the cell and the nucleoplasm of the cell, because these are actually photos of cells on the right infected with HPV. We also see changes that can be seen macroscopically with a visible light microscope in the formation of cells called koilocytes. That dark red cell in the middle that has essentially a clear zone around the nucleus of the cell is identified as a koilocyte. The halo shows a very sharp margin, and then the cytoplasm of the cell on the outside of the halo has a dark red color. Apparently this is very characteristic of a cell that's infected with HPV. Now, why the HPV results in some of these abnormal changes that we're seeing in this slide may or may not be well known at this time. And of course, if you're more interested or want to learn more, please be sure to go online or use a virology textbook and learn more at your leisure. We're not going to test on anything above and beyond some of these examples that we have here on our slides, but HPV infections are certainly becoming more important in human pathology almost on a weekly basis as scientists are discovering more and more information about this particular group of viruses. Now, to get back to our question about a vaccine that prevents cancer, there is now available 2 vaccines that can prevent infections with HPV. Gardasil has the most press, or seems to be the most commonly known, but both are all of the types of HPV vaccines. Basically all do the same thing. They prevent the virus from attaching and entering into the cells and causing many of those abnormal changes that we've just discussed in the previous illustrations. It's estimated that HPV infections are now considered to be the most common STD in the United States. And unfortunately, because of the transformation that these viruses can cause in cells, especially cervical cells, first we see dysplasia, which means the cells are growing abnormally, but don't have the extreme abnormal appearance that we might see in a benign neoplasm, where you definitely start to see transformation occurring, or stages of transformation beginning in the infected cells, all the way up to invasive carcinoma, where the cells are dramatically changed and now trying to disseminate throughout the body. There are various types of HPV. Some are more commonly associated with cervical cancer and genital warts than others. Now, HPV 16 and 18 and HPV 6 and 8 are the 4 different types of HPV that have been used in the manufacture of the HPV vaccines. For HPV 16 and 18, about 70% of all of the cases of cervical cancer are caused by HPV 16 and 18. That's a fairly high percentage, considering there are dozens of other types of HPV. HPV type 6 and 8 cause about 90% of all cases of genital warts. And the occurrence of genital warts has a high association with other kinds of pathology, including the formation of cervical cancer in patients who start out with genital warts. Now, if you want to see images of either one of those conditions, there are plenty of those images online on the internet. Notice that the incubation stage for papillomaviruses is anywhere from 3 weeks on up to 8 months after exposure. So just because you've been exposed and you don't see any symptoms after a few days does not mean you are not infected. In fact, currently, especially when women go for their annual exams or their gynecological exams, frequently gynecologists or clinicians routinely screen for HPV infection, even if there aren't any obvious signs or symptoms, or the patient history doesn't suggest immediate exposure. Now, the Gardasil and the various other HPV vaccines seem to be fairly effective, but of course the debate is they have to be given at a fairly young age prior to, ideally, exposure to the viruses. So there's been a fair amount of controversy about when to give it, who to give it to, and in any case, if we have a vaccine that can prevent cancer, would we and should we use it? It's one of those not only medical debates, but also an ethical debate. The one interesting thing about virology is there's always something new. In 1999, West Nile virus apparently entered North America. We had various other types of viruses that could cause encephalitis, but West Nile had never appeared in the United States or Canada or Central America until it entered into New York in 1999. Since then, it has essentially spread throughout the United States and is now entering into Canada and Central America. In 2002, we saw the occurrence of severe acute respiratory syndrome, or SARS, in China. This was a highly contagious, highly virulent respiratory infection, which within 2 weeks of the Chinese medical establishment finally admitting that there was a problem, was transferred from the Far East to North America through an individual who had contracted the virus, was asymptomatic, got on a plane, and flew to Canada. And of course, every year there's always some kind of new interesting influenza virus strain or possibly a newly emerging virus occurring somewhere in the world. Always a challenge and always of interest to those of us in microbiology, and more specifically, virology, which is the scientific study of viruses. Fortunately, viral infections can be effectively controlled with immunization or vaccination. In both cases, immunization and vaccination, there are subtle differences in definitions, but we're essentially doing the same thing. We're stimulating or affecting a person's immune system to simulate a disease without all of the deleterious or pathological changes that occur with the real disease. We're giving them a milder form of the disease, or we're simply exposing the patient to pieces and parts of the microorganism to trigger active immunity. Active immunity means your body made the immune factors. We call it active artificial immunity because with vaccines, we're deliberately exposing the person or animal to the microorganism. So active artificial immunity is what we're trying to develop using vaccines. And one good example, actually two now, of the effectiveness of vaccines is the eradication of two diseases, one human, one animal. Smallpox was eliminated from the world population of people and officially declared gone or eliminated from the world in 1981. Smallpox has in human history possibly caused more death than any other microorganism. There are some strong contenders, or there's some severe or pretty intense competition from a few other pathogens, but smallpox caused quite a bit of mortality and morbidity, death and sickness over the thousands of years that it had occurred in human populations. Similar thing could be said for rinderpest. Rinderpest was a very serious disease among cattle. And as some of you are well aware, cattle are the domesticated animals that we rely on for often various kinds of manufacturing products or foods. Now, recently, rinderpest has also been eliminated from the world population of animals. So, smallpox, rinderpest, both excellent examples of how effective use of vaccines can essentially completely eliminate certain types of diseases from the world. And so effective use of vaccines is very important. There are always complications, there are always possible hazards. We do have to weigh the benefit with the risk. But a good vaccine with a good track history or a good history of use and prevention of disease is a very effective means of controlling or even eliminating diseases worldwide.